So where we left off on Friday, we talked a lot about light, a lot about color, and we talked kind of an overview of photosynthesis. Today we're going to get into some of the more of the details of the reactions of photosynthesis, and it starts with having a better understanding of the chloroplast, and we did talk about this a little bit at the very end of class, but I wanted to just go over it again. So here's a typical chloroplast. Photosynthetic organisms have at least one of these. There are some unicellular photosynthetic organisms like cyanobacteria that might only have one single, well, cyanobacteria won't have chloroplast, but they have something like this. But there are single-celled protists that only have one single chloroplast. Land plants like trees will often have 100 chloroplasts per cell in their leaves, and you think how many leaves are in or how many cells are in a leaf and how many leaves are on a tree. There are lots and lots of chloroplasts on that plant. Chloroplasts have within them these stacks that are called grana. One single grana is granum, and those stacks are made of these little sacs called a thylakoid. And the thylakoid is going to be where the reactions we are going to look at today occur. The thylakoids are where pigment molecules, the chlorophyll A, chlorophyll B, the carotenoids that I talked about on Friday are embedded, and we're going to look a little closer at that. So just think about the scale of this. If you think about a tree, think about how many leaves are on a big tree, and then think about one leaf. Each of those leaves has hundreds of cells, and then each of those cells has hundreds of chloroplasts. Then each of those chloroplasts has 10 to 100 grana, and each of those grana has 10 to 20 thylakoids, and we're going to be looking at one little teeny location on that thylakoid. So we're looking at something that's incredibly small, and the scale of which is huge when you look at the whole organism. Yet there are some organisms that only have one chloroplast, but even so they still have lots of locations for photosynthesis to occur. So just kind of keep that in mind as we're going through today, that the scale is pretty gigantic. So on the thylakoid are structures we're going to be talking about in 2 slides, but before we get there, I want to talk just a little bit more about what happens when light energy is absorbed by a pigment molecule. So we said light energy that hits a pigment molecule is either absorbed or it's reflected or transmitted. Light energy that's absorbed by a pigment molecule, this is just a chlorophyll molecule that's isolated, that's not in the chloroplast, because we'll see something kind of different happens there, but very similar. What happens is the energy of the electrons within the atoms that make up that pigment molecule, when they're hit with light energy, they get excited. So if you remember atoms, so if we just think about an atom that maybe has some electrons on it, these electrons will become excited and they'll actually be kicked to a higher electron shell momentarily. So they're very highly excited. It increases their potential energy. They get kicked out to another orbital, and they're going to eventually come back down to the less excited state. And when they do that, that energy needs to go somewhere. And if you extract chlorophyll and you take it out of the plant and you shine a fluorescent light on a UV light, you give them a lot of energy, you can cause them to actually glow. And that's one of the ways they can release the energy. It's called fluorescence, and they can glow. Another way that energy can be released is through heat. So it can be released through heat, it can be released through light, and that's what would happen to an isolated chlorophyll molecule. We're going to see that when the chlorophyll molecule is in the chloroplast, that energy goes somewhere else. Same thing happens when light hits pigments in either your skin or pigment molecules in your clothing. Pigment molecules in your clothing, it typically gets converted to heat, and this is why you feel your clothing gets warm in the sun. Same with your skin gets warm in the sun. If you've ever worn like glow-in-the-dark, anything that glows in the dark, right? Kind of like t-shirt that might have glow-in-the-dark paint on it. What's happening is that that energy is actually being absorbed by those pigment molecules, and they can hold on to them for a little bit. And then when you're in a dark room, they'll still be releasing that energy, and they'll be releasing it as light. And that's what glow-in-the-dark paint does. It just slowly releases it as light, which is kind of cool, right? So that's what's happening when energy hits a pigment molecule. Is it excites the pigment molecules temporarily, and then the pigment molecule will release that energy in a slightly different form. Okay, so pigment molecules are not just freely floating in the plant. They're attached to something, and what they're attached to is something called a photosystem. So the thylakoid membrane is covered in photosystems, and photosystems are locations where chlorophyll A and accessory pigment molecules are bound. Photosystem is a protein complex, which means it's not just one single protein, it's many proteins kind of stuck together, and those pigment molecules are bound to the proteins that are embedded in the thylakoid. Most pigments are what are called antenna pigments, and they're located on the outside of the photosystem. So here is the proteins on kind of the periphery. This is a cross section, so it's really, there's a circle of proteins that make up kind of the antenna complex, the antenna proteins, and they have the antenna pigments on them. And then in the very center of the photosystem is another protein that's called the reaction center of the photosystem, and it has a pair of chlorophyll molecules embedded inside of it. So most of the pigment are on the periphery, most of the pigment molecules on the periphery of the photosystem, but in the reaction center there's just a pair of chlorophyll molecules. And what happens is light is going to more than likely hit pigment molecules that are part of this antenna complex. So just like a radio antenna, that it's kind of sticking out and it's absorbing radio waves, these antenna proteins are designed so that light will hit them and will hit the pigment molecules in the antenna complex. And so as light hits those pigment molecules, it's going to excite the electrons in the pigment molecules, just like we were talking about on the last slide. When those electrons relax back down to the unexcited state, that energy is going to be released. And what happens is it shuttles the electron, or the energy, not the electrons, shuttles the energy to a neighboring pigment molecule. So energy that was absorbed by this outside pigment molecule is transferred to the next pigment molecule over. It excites that one. And again, when that one calms back down, it releases its energy and passes it on to another neighboring pigment molecule. And eventually, that energy makes its way to the center, to the central chlorophyll molecules. And imagine the same thing is happening elsewhere. So the same thing is happening here. Light hits this pigment molecule, that energy is transferred and funneled, basically funneled in to the central chlorophyll molecules. So since there's all these pigments surrounding those chlorophyll molecules, they are funneling all the energy that is hitting them into the center. And so there's a massive amount of energy that's being concentrated on this central chlorophyll molecule. Okay? That's how a photosystem works. Photosystem funnels in the energy that's captured by the antenna complex into the center, into the central chlorophyll molecule that's at the base of the reaction center. Okay? That's the capture of energy. And we're going to see what happens to that energy at that point, but it's a massive amount of energy that is funneled in. Okay, so just an overview, again, of the two stages of photosynthesis, and we're going to talk about the light reactions in a fair amount of depth today. Photosynthesis consists of the light reactions and the carbon cycle, or the Calvin cycle, sometimes called. Here, the light reactions occur on the thylakoid. We're going to go into more depth of those today. The light reactions are going to absorb light splitting water, releasing oxygen. They're going to generate ATP by a process called photophosphorylation, and we'll talk about that in a little bit of detail. And they reduce NADP to NADPH. And reduction, remember, is gain of electrons, so they're going to transfer electrons to a molecule that's called NADP plus and convert it into NADPH. So it's also going to pick up some hydrogen as well. And then we'll see later on that ATP and NADPH are going to power the carbon reactions, the Calvin cycle, to convert carbon dioxide into sugar. So I'm trying to use the terms that are in your textbook. The carbon cycle's what it calls it. If you read about this anywhere else, you'll probably see the word Calvin cycle. A lot of these things are named either by the molecules that are involved in them. This one takes in carbon in the form of carbon dioxide to make a carbon-based molecule called sugar. But they're also named by a person that discovered them. And Calvin is a name that is associated with this reaction because of his involvement in discovering this. Okay, so today we're going to talk a lot about the light reactions, this process that splits water, releases oxygen, and generates two power sources, ATP and NADPH. Okay. So in the light reactions, we're going to see there are two different kinds of photosystems. They're both the same concept as what I talked about on the last slide. They both funnel energy into a central pair of chlorophyll molecules. But they have slightly different things that they ultimately do with that energy. And so we'll talk about them as distinct photosystems, even though their structure is not drastically different. There's photosystem 1 and photosystem 2. Photosystem 1 was discovered before photosystem 2. So that's how it got its number. But in terms of the chain of events that's going on in the light reactions, photosystem 2 is going to function first. So they function in kind of reverse order. There are actually some photosynthetic organisms that only have a photosystem 1. They don't have this second photosystem. So that's another reason for the numbering system. Okay, so photosynthesis begins with light energy striking pigments in photosystem 2. And like I said before, the energy is transferred from those antenna pigment molecules, into a central chlorophyll A molecule. And that chlorophyll A molecule is often called P680 because 680 nanometer energy is what it absorbs the best. So the energy that is fluorescing off of these chlorophyll molecules from the antenna chlorophyll molecules a lot of it is at 680 nanometers of energy. You don't really need to know that. It just helps to distinguish the two photosystems by using something to name their chlorophyll molecules rather than saying the chlorophyll molecule at the center of photosystem 2. I can just say P680. Okay, so all that energy is being funneled in to P680. And remember that when energy hits the atoms within a pigment molecule, it causes electrons to become excited and to jump to outer orbitals. The photosystem is designed to hit that chlorophyll molecule with a massive amount of energy. So much energy that when it excites the electrons in that chlorophyll molecule, it excites them so much that they actually get released. Instead of just jumping to another orbital, they get kicked off of the pigment molecule. So the electrons from P680 will actually be kicked off of P680, and they're going to be transferred to the first protein in an electron transport chain that we often call the primary electron acceptor. So this might be, so this right here, the electrons here are going to be kicked out and they're going to be sent to an electron acceptor, which starts something called the electron transport chain. So when P680 when it loses an electron, we start calling this P680 plus, as it's lost an electron, so it's lost a negative charge, so it's now positively charged. So P680 plus is not a very useful molecule. It's lost an electron. If it gets hit with more energy, it's not going to lose any more electrons at this point. So it needs to be recharged so that it can fire again and we can continue doing the photosynthesis process over and over rather than just use this photosystem once. So it needs a steady supply of electrons to convert it back from P680 to P680 plus to P680 again. And what it gets its electrons from is water. So water will donate electrons to reduce P680 plus back to P680 as water is split into hydrogen and oxygen. So here's water being split into hydrogen and oxygen, and electrons from water are being transferred to the chlorophyll molecules that make up P680. So remember I said it's a pair of chlorophyll molecules, so it's 2 electrons, and convert it back to P680. And this allows it to fire again. So again, energy can be funneled down to P680. It will lose electrons, becoming P680 plus. Water will donate electrons, bringing it back down to P680. So it's constantly, water is constantly supplying photosystem 2 with a steady stream of electrons. And ultimately what's happening is light energy is exciting those electrons and kicking them into an electron transport chain. Okay, does this make sense so far? What's going on? So light energy funneled in, excites electrons, they get passed off, and they're now energized, excited electrons, and those electrons are replaced by the splitting of water. You have a question?
Electrons from P680, they go to the
electron transport chain, yeah. Yeah, so some texts will say primary electron acceptor is their first spot, but really that's the start of the electron transport chain. So they're going to an electron transport chain. Okay, and they're energized now. Okay. So this is the first step. This is actually a super cool process because without this, we wouldn't exist. This is how a plant is able to capture energy. So we've now converted light energy into an energized electron. Okay. So that energized electron is now going to do work. It's now got a lot of potential energy. It's going to do some work for us. Okay, so the next location that that energy goes, that electron now will travel through an electron transport chain. And we talked about this briefly in an earlier chapter. An electron transport chain is just a series of proteins that will go through redox reactions. So they will accept an electron, so they'll be reduced, and then they will pass on that electron. They will be oxidized. So they're constantly accepting an electron and then passing it on, accepting an electron and passing it on. That's what an electron transport chain is. As those proteins accept that electron represents stored energy, and those proteins can actually use that energy to do something. And what they're going to do is they're going to use the energy from the charged, energized electrons to drive active transport. And they're going to take hydrogen that was out in the stroma, out in the fluid, and pump it into the thylakoid space. So those thylakoids are these little sacs, remember, that are stacked on top of each other. And so hydrogen ions are going to be pumped from the outside of the thylakoid into what we call the thylakoid space, which is the region inside of those sacs. Okay? So energy from photosystem 2, energy that was captured by photosystem 2 and converted to energized electrons, those energized electrons will power proteins in the electron transport chain to actively pump hydrogen into the thylakoid space. So if we're using active transport to pump hydrogen in here, which side of the thylakoid membrane do you think has a higher concentration of hydrogen? All right, yeah, the inside. So otherwise, we would just open up a channel and it just flow in, right? So we're building up a high concentration of hydrogen on the inside of the thylakoid. We're actively pumping hydrogen into the thylakoid. So this is creating membrane potential. It's creating a large differential between the inside and the outside of the thylakoid. Okay, so if I was to open up a channel here, where would hydrogen go? Out. All right, it would go out, yeah. So hydrogen would just flow out. So just like water going over a dam if you open up a port in the dam, right? But instead of just letting it flow out freely, we make it flow through an enzyme called ATP synthase. So hydrogen concentration gradient is formed across the thylakoid membrane. And then instead of just flowing out through a transport protein, it's going to flow through an enzyme called ATP synthase. And as it does, it's going to power phosphorylation of ADP. So it's going to combine ADP with ATP, or ADP with phosphate to make ATP. So ADP is converted into ATP using the energy of diffusion through ATP synthase. Now, there's actually a really cool way that ATP synthase works. ATP synthase isn't just a big purple blob like this. It actually acts like a turbine. As hydrogen flows through it, it actually physically turns proteins, and that kinetic energy of motion, of the motion of that enzyme, is going to help catalyze this endergonic reaction of phosphorylating ADP. So this process right here is called photophosphorylation because it's using the energy from light to phosphorylate or add a phosphate to ADP to make it ATP. Okay, so does this make sense so far? Okay, so active transport, build up concentration gradient, that potential energy of diffusion is captured by ATP synthase to generate ATP. So just like water going through a turbine in a dam. Okay, so we've now used up our electron energy going through the electron transport chain basically to generate ATP. So we've made one of our two products. We've made ATP. There's one other energetic product that light reactions make, and that's going to be generated by photosystem 1. Okay, so just like in photosystem 2, light energy is captured by photosystem 1 and funneled down to central pair of pigment molecules in photosystem 1. In this case, photosystem 1 captures energy at a slightly different wavelength, 700 nanometers. So we call those central chlorophyll molecules P700, but you can also just call them the central chlorophyll A molecules of photosystem 1, and that's fine as well. So energy is transferred to P700. P700 will release energy to another electron transport chain here. And P700 now, that becomes P700 plus, in order for it to regain electrons, rather than splitting water again, it's just going to take electrons that are coming off of that electron transport chain that generated the ATP. So there's low energy electrons that are coming from photosystem 2 through that electron transport chain. They're going to replace the electrons in P700 plus, converting it back to P700 again. Okay, but other than that, pretty much exactly the same as photosystem 2. Energizes electrons. Remember, photosystem 2 got its electrons from water. Photosystem 1 gets its electrons coming off that first electron transport chain. Okay, so far? Okay, so where do those electrons go? Electrons from P700 are going to go through another electron transport chain and then be transferred to NADP plus reductase, so an enzyme called NADP plus reductase, that is going to reduce NADP into NADPH. So electrons plus hydrogen plus NADP is going to make NADPH. And NADPH is an electron carrier. It's going to carry those electrons, which still have a lot of energy in them. So we haven't lost any of the energy from these electrons. The energy of those electrons is going to be brought to the next part of photosynthesis, the carbon cycle. Okay. So photosystem 2 produces primarily ATP by powering an electron transport chain to drive active transport. And photosystem 1 primarily produces NADPH by energizing electrons and then transferring them to NADP, converting it to NADPH. So we now have ATP and NADPH available for the next step of photosynthesis. Okay. So that is the light reaction. So I'm going to go over it again. This is definitely something I expect you to have a good understanding of. This is the key concept. This is really the reason why life can exist, is because there are organisms that can do this process. So, and this is probably, I would say, the most important step. It's actually converting light energy into a chemical energy of ATP and NADPH. So I would expect you to understand the light reactions and this process of energy flowing through the different photosystems and the electron transport chains. Okay, so here's an overview again, and then if you have any questions, ask them at this point so we can go over that again. So this is what the whole thing looks like. If you zoomed out a little bit, here's the thylakoid. This is one of these sacs. So this would go around and become a whole kind of sac, and this is a cross-section of it again. So here is photosystem 2. So light energy hits the pigment molecules in the antenna region of photosystem 2. That light energy is funneled down to the central chlorophyll molecules, P680, and excites the electrons in P680, kicking them off, sending them to an electron transport chain. Those electrons are replaced by electrons coming off of water, as water is split into oxygen and oxygen is released. The high energy electrons from photosystem 2 will pass through an electron transport chain. As they do, they're going to power the active transport of hydrogen from the stroma into the thylakoid space, building up a concentration of hydrogen inside the thylakoid space. Hydrogen is then going to diffuse through ATP synthase to generate ATP. Okay, in photosystem 1, light energy again hits antenna pigment molecules and is funneled down to the central chlorophyll molecules, P700, exciting those electrons, sending them off to NADP reductase, and those electrons are replaced by electrons coming off of the first electron transport chain here. The high energy electrons that have been passed to NADP reductase are going to reduce NADP plus into NADPH. Okay, so we now have ATP generated here through diffusion through ATP synthase, and we have NADPH that's generated by NADP reductase from the energy from photosystem 1. Okay. Questions on that? I think you'd be able to explain that. All right.
On that point, yeah.
All right.
What exactly is NADPH?
It's an energetic compound. It's an electron carrier, we call it. So we're going to see it basically is like ATP. It's a source of energy. It's going to provide energy and provide electrons to the next step of photosynthesis. So, and we'll see them again in respiration, something similar. They're just intermediate energy carriers. Okay, yes.
I'm sorry, can you just repeat what happens from the electron from photosynthesis one,
Photosystem, yeah.
photosystem to the next?
Okay, photosystem 1 electrons. So the electrons are excited. They're passed NADP reductase, and they are then transferred to NADP, reducing it into NADPH. So NADP plus hydrogen plus electrons gives you NADPH, the electron carrier. So just like ADP is the lower energy form and ATP is the high energy form, NADP is a low energy form, and NADPH is a high energy form. So NADPH represents stored energy. Okay? So I'll go over it one more time. This is something that's important that I think you should be able to explain. So light energy hits photosystem 2, pigment molecules. That energy is captured by the pigment molecules and transferred to a central pair of chlorophyll A molecules, P680. It excites the electrons in P680, sending them to an electron transport chain. Those electrons are replaced by electrons coming off of the splitting of water, where oxygen is being released as a byproduct. Electrons coming off of photosystem 2 head through an electron transport chain. They drive the active transport of hydrogen into the thylakoid space. That buildup of hydrogen creates membrane potential and allows for diffusion through ATP synthase to generate ATP. In photosystem 1, again, energy is absorbed by pigment molecules in the antenna complex. It's funneled down to the central chlorophyll molecules, P700, exciting those electrons and ultimately sending them to NADP reductase to convert NADP into NADPH. And electrons of P700 are replaced by electrons coming off of the electron transport chain. Okay, so now we have ATP and NADPH. And that's the first half of photosynthesis. Okay? So definitely review that. All right? Okay, so let's see how well you were paying attention. So which one of these is the best answer for how do the light reactions produce ATP? So here's what you said. And the 67% of you are right. So it's, whoops, sorry. It is potential energy stored in a hydrogen ion gradient is used to synthesize ATP. So where ATP is synthesized by hydrogen diffusing through ATP synthase, right? And the reason why hydrogen diffusing through ATP synthase can power something is that there's a high concentration of hydrogen inside the thylakoid and a lower concentration outside. That's built up by the pumping of hydrogen into the thylakoid space, which is powered by photosystem 2. So it's not that photosystem 2 directly adds a phosphate. It doesn't. Photosystem 2 energizes electrons. Those electrons power active transport, which pumps hydrogen across a membrane, and then that diffusion leads to ATP synthase. Energy released by electrons directly used to synthesize ATP. That's not true either, because the energy released by the electrons, what does that directly power? The active transport, right? So it's active transport builds up hydrogen ions, and then the diffusion of hydrogen is what synthesizes ATP. And photosystem 1 doesn't have, at least as far as I've taught you, anything to do with this. There are exceptions. Some plants only have photosystem 1, and there's actually another route for electrons, but we're not going to talk about it, so ignore that. But still, that wouldn't directly do anything anyway. So it is kind of a really roundabout way to make ATP. You're using energized electrons to pump hydrogen, and then hydrogen diffusing is generating ATP. So it's like you were using a wind turbine to power a pump to pump water up to the top of a dam, and then having the water go through turbines in the dam to generate ATP. to generate electricity. So it's a very weird kind of way that the cells do this. And you'll see the same thing happens in mitochondria, but it works. It's building up a concentration gradient, which ultimately the concentration gradient is what drives ATP synthesis. So does this make sense? Those of you that didn't get this right, or those of you that at first didn't get it? Okay, what's confusing about it? Okay, you do. Okay, all right. So the big thing is it's a concentration gradient that ultimately makes ATP. It's not directly off of the energy. It's something else. Okay. Okay, so I'm just going to, I only have a little bit of time left, so I'm going to introduce the carbon cycle, and we'll do more of this, and I'll show you a video on Wednesday of this. So the carbon reactions, also called the Calvin cycle, if you look this up in any other text or if you look this up on YouTube or something like that, the carbon reactions are what actually produces sugar molecules. They're what actually produces carbohydrates. The Calvin cycle is going to use the energy that we just created, ATP and NADPH, to build sugars from smaller molecules. Ultimately, just to kind of talk about the context of this, so ultimately those sugars that are produced by the plant, what are they going to be used for? Right, energy for what? All right, so the plants are producing them so that we can eat them and be energized. They're really benevolent and they're just producing sugars. Not really, right? What are they making that energy for? So they can sell it at the market, like that, right? No, they're using it for themselves. So why do you think a plant makes sugar rather than just driving cellular processes off of ATP that it produced in the light reactions? What would be the benefit of making sugar? Okay, yeah, bingo. So why do we store fat? All right, someday you might not be able to eat and you want to be able to live for that day as well, right? So you can live off of stored energy. Most of us don't have that problem of not having food, so we just keep adding more and more fat. But that's the whole idea, is that you're storing for later. And that's what plants do, because there are times when there's no light, like night. That seems to happen every day. And like when there's storms, or like the winter, when there's less light. So it's like kind of the equivalent of one of those solar-powered calculators that just runs directly off of the solar power versus something that has a battery in it. If you put your thumb over the solar panel on the solar-powered calculators, it stops working, right? And plant doesn't want that to happen. It doesn't want to die when the sun goes down, right? It wants to be able to survive under many conditions. So it produces sugar through the Calvin cycle so that it can then break down that sugar to generate ATP later on and drive its processes off that ATP. So it's like having a battery that you charge by solar panels, and then you run the house off of that battery. Okay, so the Calvin cycle is the process of making that storage form of the fuel. Carbon is going to enter this cycle as carbon dioxide, and it's going to leave as a 3-carbon sugar called pGal. So PGAL. Eventually, that 2 pGals can be linked together to make glucose. So glucose is a 6-carbon sugar, but we're going to really only look at it up to the point where we make a 3-carbon sugar, and then we're going to just say, well, pGal can be made into lots of things like glucose, like fructose, like any other kind of sugar. The Calvin cycle is going to have 3 different stages to it. It's going to have an initial stage where carbon is fixed, and carbon fixation is the process of taking in carbon as carbon dioxide and converting it into an initial chemical compound, so sugar compound. And that process requires an enzyme And the enzyme that is hugely important for the large majority, and really almost every single plant there is something called rubisco. So we're going to talk a little bit about rubisco on Wednesday. The second step of the Calvin cycle is the synthesis of pGal, so converting this initial carbon-fixed product into pGal. And then the last step of the Calvin cycle is to regenerate a molecule called RUBP, which was what initially bound to carbon dioxide in the first place. So we're going to see that the Calvin cycle, it's called a cycle for a reason, you're going to end up where you started. And it's going to continue going over and over and over again in a cycle. And as it does, every time it goes through a cycle, a little bit of pGal is released. So carbon comes in, pGal is released, and then it goes back and regenerates what it started with. Okay. So I'm not going to go into the details of this today. I'm going to stop here, and we'll go into the carbon cycle in a lot of detail on Wednesday. All right?